Hello everyone, my name is Ravinder Singh and I'm currently working as a technician in Dr. Phyllis Coxell's lab at the University of Manitoba. Today I'm going to present our recent research on extrusion processing for the manufacturing of texturized vegetable proteins from major Canadian pulses. As we're moving towards more sustainable protein sources, pulses are going to play a major role in meeting the protein demands for the future. Pulses have a number of advantages over animal protein sources, such as their less carbon footprint and less water utilization, and also the consumption of pulses have been associated with several health benefits. However, at the same time, pulses have several drawbacks, such as the presence of anti-nutritional factors and not being the complete protein sources because of limiting sulfur-containing amino acids. And also, some pulses have flavor issues associated with their use. Most of these drawbacks can be addressed with the use of proper processing techniques. And in this research, we are using extrusion processing for the purpose of value addition to the pulse protein isolates. Extrusion is a high temperature, short time processing technique that is commonly used to produce a variety of food products such as puffed snacks, food ingredients, meat analogs, and texturized vegetable proteins, which are the focus of this research. Texturized vegetable proteins are basically the extruded products which mimic the fibrous structure of meat products when rehydrated, and they are commonly used as meat extenders in the food industry. Inside the extruded barrel, the protein molecules first gets denatured because of high temperatures, and when this denatured protein melt travels further along the extruded barrel, the alignment of protein molecule takes place and some cross-linking occurs, which then give rise to the formation of fibrous structures. However, the fibrous structure formation is not as simple as this because several extrusion processing parameters need to be optimized to get the best possible structure. And this is what we are doing in the first phase of this research. We are trying to optimize the feed moisture content, dye temperature, and secure speed to produce TVPs from pea, lentil, and fava bean protein isolates. After that, we are analyzing them for their physical, techno-functional, textural, and structural properties. And our ultimate goal is to commercialize these TVPs as meat substitutes after further nutritional enrichments and sensory analysis. I'm glad to present some of our initial results from pea protein TVPs. By looking at the scanning electron microscopy images of these TVPs, we can see that the TVPs obtained at 130 degrees Celsius dye temperature showed some fiber-like elongated structures, which were not there at lower dye temperatures. So we can assume that higher dye temperatures might be better for pea protein TVP's production. It is important to see if this fibrous structure also translated in their properties. So in this table, we can see that these TVPs, those were produced at 130 degrees Celsius, had comparatively higher water holding capacity, lower hardness, and higher saprophogenous values. And all these attributes are desirable in the texturized vegetable proteins. So what significance this research has for the overall protein ecosystem? First of all, we are adding value to major Canadian pulses, and Canada being one of the major producer of pulses, this research will have immense significance to the Canadian protein industries. We are also diversifying the current TVP space in which soya protein products are dominant. At the same time, we are also gathering comprehensive information about the technofunctional properties of different pulse proteins, which will help in producing more sustainable food ingredients in the future. With that, I would like to acknowledge our collaborators and our funding source. And thank you very much.